Genetic engineering. I want to show the example of genetic engineering following the OCR A-level specification. If you're using their endorsed textbook, you'll find the information that I'm going to go through on this PowerPoint on pages 228 to 230. Now the example I want to use to explain to you how genetic engineering is done is the production of human insulin from bacteria. Now to be able to genetically engineer bacteria to produce human insulin, we need to actually give them the actual gene for insulin. Now all of the cells in your body will contain this gene, but only one group of cells will actually be expressing that gene, and that's going to be inside the pancreas, in particular the beta cells in the islets of Langerhans. So you need to take those cells, and now they're going to be expressing a gene, which means there's going to be a lot of transcription and translation going on, particularly transcription of the insulin gene into messenger RNA. So most of the messenger RNA inside these cells is going to be the information to make the human insulin. So once the messenger RNA has been obtained, we need to now convert that into DNA, and that can be converted using an enzyme called reverse transcriptase. So this is transcription in reverse. Now I'll show you a little bit more about that in a minute. So the reverse transcriptase will complementary insert DNA nucleotides now, but this is going to be single-stranded, so we need to make this double-stranded, so we have a double helix, and this time you can use the enzyme that you would have looked at already, which will be DNA polymerase. A primer will need to be added first, but DNA polymerase will run along to make the other complementary strand of the DNA double helix. And what we're left with is the double helix of DNA, which is called cDNA. Some people think this is copied DNA, but the C actually stands for complementary DNA. Now that should be the insulin gene, and that's what we can now insert into another organism to genetically engineer it. Before that, a few other points on this particular part. And if you are using the OCR endorsed textbook, please have a look at page 228, 229 for this. Now, as I mentioned, reverse transcriptase comes from viruses, particularly retroviruses. These are the ones that insert RNA into cells and not DNA. And so what they also do is they have to get their RNA converted into DNA so it can go into the host cell genome. So they also provide information for the reverse transcriptase enzyme to be synthesized. HIV is such a virus that is a retrovirus and therefore possesses reverse transcriptase. I also mentioned that DNA polymerase will also require a primer to make the single-stranded DNA into a double helix. And there are other methods that we can get the genes as opposed to using the messenger RNA. One of the ways is that if you know the actual sequence in the first place, just artificially synthesize the gene. There are machines that will put the correct nucleotide in the correct sequence for you. Also, if we know where the gene is and we know a little bit about it, we can use primers to bind either side of the gene and then use polymerase chain reaction to make many copies of that particular gene. And finally, if we don't know where the gene is but we know something about it, use a DNA probe to locate the actual gene in the genome, and providing that there are restriction enzyme sites, which I'll say a little bit more about later, we can cut that gene out directly. Anyway, back to making bacteria genetically engineered to produce insulin. Once we've obtained the gene, we actually have to put it into the bacteria, and it's not an easy process. So what we use is we use something called a vector. An example I'm going to show you as an example of a vector is plasmids. I'll say a little bit more about plasmids shortly. Now plasmids are circular DNA, and what we can do is that we can cut open the plasmid using restriction enzymes, and once the plasmid has been cut open, we can stick our gene, in this case the human insulin, into the bacteria. Before we do that, a little bit more about plasmids. As I said, they are circular DNA which are found in bacteria, and the good thing about this is that as bacteria replicate, the plasmids also replicate. So all the new bacteria formed in next generation and so on will also contain this plasmid. And if we've got the gene in there, the human insulin gene, we want all the next generation and subsequent generations to contain that plasmid with the human insulin gene in. So it's very useful in genetic engineering as a vector. And just to explain again what a vector is anything that can carry something from one place to another. So our plasmid is going to carry our gene, our human insulin gene, into a bacterium. Another vector that you would have been familiar with from a previous chapter would be, for example, mosquito, which transfers the malaria parasite from humans to humans. Right, so we need to put our gene into the plasmid. Now, if we were to let the gene and the plasmid mix together, it's unlikely they would actually stick to each other. So what we need to do is we need to stick or put on sticky ends which complement each other. Hopefully you understand this as you see the video. So sticky ends are going to be a sequence of bases. So on one end we might put a string of C's, cytosine, and the other end a string of guanines. We also need to do that to the gene that we wish to insert into the plasmid. So in this particular case, a string of C's and a string of G's. Then, once we mix the plasmid and the gene together, you can see now that the complementary sticky ends will bind to each other, so you'll get hydrogen bond formation between the cytosines and the guanines. This will help to stick the gene into the plasmid. Now, to make sure that we've got a good join, the sugar phosphate backbone will need to be joined up, and for that we use another enzyme which will be DNA ligase. You'll find a description of that on page 229 of the OCR endorsed textbook. What we've got now is we've stuck a gene from one organism into a plasmid, and this means that we have mixed up the DNA, recombined it, so we give this the phrase recombinant DNA. Right, at this point we'll just take a little pause and think about what restriction enzymes are now, because I've been mentioning those several times and we've seen them in action. They're also called restriction endonucleases, and they are obtained from many types of bacteria. And the reason why bacteria contain these DNA cutting up enzymes is because they are susceptible to infection from little viruses called bacteriophage. Bacteriophage will inject their DNA into the bacteria, and the bacteria will make lots of copies of the bacteriophage, and there's a picture of a bacteriophage on the video now. So as I said, the bacteriophage will bind to the membrane of the bacteria and insert its DNA. And as the bacteriophage are made within the bacteria, they will burst out of the bacteria, effectively killing it. So in defense, bacteria have these restriction enzymes which will cut up the DNA of the bacteriophage. I should mention at this point, bacteriophage are of potential interest in medicine. Because of the increase in risk of antibiotic resistance, we need other ways to destroy bacteria infections. And one possible way is to use bacteriophage, which won't harm eukaryotic cells at all. They'll only infect bacterial cells. So this could be a potential source of treatment for bacterial infections. Now, restriction enzymes have particular names, and in the textbook for OCR you'll be reading about EcoR1, and it's called EcoR1 because it was obtained from E. coli. So E for E. coli, Escherichia, the genus name, and the co bit from the species name, Eco. And the particular strain that this enzyme was first identified in was the RY13 strain. There are many different strains of E. coli, and because it was the first one, it's given a destination 1, so eco R1. There are many other types of restriction enzymes, and they all have names based upon the type of bacteria that have been identified in the strain. Okay, so restriction under nucleases cut at specific DNA sequences, and this is really important. The DNA sequence you're looking at now I've highlighted in red, and these sequences tend to be palindromes and quite short, 4 to 6 base pairs long. So palindromes because they read in both directions, the same sequence. Now sometimes we're going to get sticky ends, sometimes we get something called blunt ends. That's where the restriction enzyme would just cut straight down the middle, and you wouldn't have any overhanging nucleotides. But in this particular case, eco R1 will leave sticky ends, and these can be used to help the insertion of the gene into the plasmid, which I'll show you a bit later. So come back to inserting the gene into the plasmid. We have to cut the plasmid open using restriction enzymes. And then we can insert the gene. Now the red bits I'm showing you here are meant to represent the sticky ends, and they should overlap. So we get complementary base pairing. And then to join up the sugar phosphate backbone, you need the enzyme DNA ligase. And this gives us our recombinant DNA, and the vector is now ready to be inserted into the recipient cell, in this case a bacteria cell. Now if we haven't got overlapping sticky ends, maybe if you've used a restriction enzyme that cuts straight down the middle, sticky ends can actually be added. So a string of C's may well be added. You can use any other bases as well, and these are joined up by an enzyme called terminal transferase. So back to the method to genetically engineer a bacteria. We've prepared our plasmid after we've obtained the gene. We've now got recombinant DNA, and it's simply a case of the bacteria taking up the plasmid. And that will be our genetically engineered organism. Now there are other ways of getting vectors into the cell. The method I've shown you is the bacteria take up the plasmid. Now to help that happen, you'd use something called heat shock, which is where you alternately change the temperature between 0 and 42 degrees. And you also add calcium ions, or calcium chloride ions. The calcium, which are positively charged, will surround the negatively charged DNA and the phosphates of the membrane and stop the DNA being repelled from the phosphates in the membrane. And that will help the DNA therefore to get through the membrane. Other methods include something called electroporation, where high voltage is applied to the membrane which disrupts it to allow the DNA to enter. Something similar is electrofusion using electrical fields. We can use transfection, and again here we use bacteriophage. Now I've already explained to you that bacteriophage will insert DNA into a bacterium. So if you've put the gene into the bacteriophage may then insert the gene into the bacterium instead. And sometimes we want to insert the gene into plants. So once you've gone to the stage of putting the gene into a plasmid, the plasmid can then enter a particular bacterium called Agrobacterium tumefaciens. Now these are bacterium that enter plant cells, and this way you've then carried the gene into the actual plant cell, if that was the intended cell. Sometimes plant cells won't take up the Agrobacterium tumefaciens, so we can use something called a gene gun. Now a gene gun is where little particles of gold or tungsten are coated with the gene and literally shot into the cells. Now, back to our process of producing genetically engineered bacteria to make human insulin. Once the bacteria got their plasmid in, they will divide, and as I said, the plasmid also replicates with the bacteria, and all the other bacteria that are produced will therefore contain the plasmid with the insulin gene. It's not as clear cut as this. The main problem is that not many of the bacteria will actually take the plasmid up in the first place, despite all the methods I've just described to you previously about getting the vector into the cell. What you don't want to do is to start growing bacteria that do not contain the gene. But there is a method to solve this problem. So as I said, we want bacteria that contain the plasmid or the recombinant DNA. What we don't want are any bacteria that don't actually contain the plasmid in the first place. They will grow in the agar, but we don't want them to. Likewise, some bacteria may take up a plasmid that isn't actually carrying the gene we want, so it would be a non-recombinant plasmid. We don't want those either, and they will also grow in the agar. What we want are those that contain the plasmid which is recombinant. So what's the technique? The technique is called replica plating, which I'll explain to you now. Plasmids contain many other genes, and some of these may well be genetically engineered, but in this particular example, this plasmid contains a gene to be resistant to the antibiotic called ampicillin. Some of you may well have had that antibiotic. So any bacteria that contains this plasmid will not be killed by the antibiotic ampicillin. They also contain another gene called tetracycline, resistance gene, and tetracycline is also another antibiotic. Now this may seem a bit strange. We're trying to make bacteria that are antibiotic resistant, but this is only for laboratory purpose to help us identify the bacteria that contain the plasmid with the insulin gene. Now, what we want is to insert the human insulin gene into one of these antibiotic resistant genes. So hopefully there's going to be a restriction enzyme site there which we can make use of. So if we use a restriction enzyme, we insert our gene. Any sticky ends there will complementary pair with each other, but to make sure of a good seal we need to use DNA ligase to join up the sugar phosphate backbone. And then what we'll have, although you can't physically see these, we'll have a number of plasmids that contain a gene, but we'll also have a number of plasmids that don't contain a gene. Then we mix the bacteria with the plasmids. Now the bacteria should take up, some of the bacteria should take up the plasmids, particularly if we use heat shock and calcium chloride ions, but the success rate of this is very low. It's a very low percentage. However, if you think about it, we actually only need to have one bacteria because those bacteria will divide anyway. We just need to be able to identify the bacteria that are taking up the plasmids. So what we do is that we after incubating the bacteria, the plasmids, you then put the bacteria onto a petri dish containing agar with the ampicillin antibiotic in. Now any bacteria that did not take up the plasmid will not be able to grow. So what we will see growing on these plates are bacteria that have taken up the plasmid, whether it's non-recombinant or recombinant. Then what we do is we do the replica plating bit. So using a velvet cloth, you press it down onto the petri dish containing the bacteria, and then press that onto another petri dish with agar, but this time the agar contains the antibiotic tetracycline. We look from above. Now, any bacteria that contain the recombinant plasmid, that's the plasmid with the insulin gene, means that the tetracycline resistant gene is not going to be expressed. So those bacteria will not be able to grow. Now that means we can go back to the ampicillin plate and we can now identify those bacteria that contain the recombinant plasmids. And these can be literally picked off and then put into more agar or nutrient broth and grown. And that way you get a pure culture of bacteria that has been genetically engineered containing the recombinant plasmid with the human insulin gene, and these bacteria will then express the human insulin, which can then be manufactured for therapeutic purpose. Right, that's genetic engineering.